Hello everyone, today we'll discuss the part 10 of the important notes for the biochemistry so that you can easily able to answer the one-liners and the MCQs in your exam. First of all, the gluconeogenesis pathway. Now, this particular gluconeogenesis pathway occurs in both mitochondria and the cytoplasm. It basically starts from the mitochondria. Now, the organs performing the gluconeogenesis, over here remember 90% occurs in the liver, predominant one, and 10% in the kidneys. Gluconeogenesis occurs during the fasting or the starving condition and also in the diabetes mellitus patient. First step of the gluconeogenesis is conversion of the pyruvic acid to the oxaloacetic acid by the pyruvate carboxylase enzyme. Okay, over here this is the anaplerotic reaction. Now, what are anaplerotic reactions? Now, such reactions are chemical reactions that forms the intermediates of a metabolic pathway. Now, acetyl-CoA is very important allosteric activator of the pyruvate carboxylase. Oxaloacetic acid produced in the mitochondria gets converted to malate by mitochondrial malate dehydrogenase enzyme, as oxaloacetic acid can't cross the inner mitochondrial membrane. Malate crosses the inner mitochondrial membrane and is again converted to the oxaloacetic acid by the cytoplasmic malate dehydrogenase. Now, if you remember, we discussed about this in the malate aspartate shuttle, right? PEPCK or the phosphoenolpyruvate carboxykinase converts the oxaloacetic acid to the phosphoenolpyruvate, which gets converted to the fructose 1,6 biphosphate. Enzymes which are common in the glycolysis and the gluconeogenesis pathway are enolase, phosphoglyceromutase, phosphoglycerate kinase, glyceraldehyde 3-phosphate dehydrogenase, and the aldolase A. All the bypass reactions of the gluconeogenesis are irreversible. Gluconeogenesis is not just the reverse, or we can say the reversal of the glycolysis, as three enzymes are different in the gluconeogenesis. Instead of the pyruvate kinase in the glycolysis, enzymes used in the gluconeogenesis are pyruvate carboxylase and the phosphoenolpyruvate creatine kinase. Instead of phosphofructokinase 1, fructose 1,6 biphosphatase enzyme is used in the gluconeogenesis. Instead of hexokinase, glucose 6-phosphatase is used in the gluconeogenesis. Glucose 6-phosphate of the hepatocyte is not acted upon by the glucose 6-phosphatase as soon as it is formed, because glucose 6-phosphatase enzyme is present in the membrane of the endoplasmic reticulum, not in the cytoplasm. Sequence of the compartments for the gluconeogenesis, very important, that is first of all the mitochondria, then cytoplasm, and then the endoplasmic reticulum. So now you might be getting the previous point over here. Glucose 6-phosphatase is common enzyme in gluconeogenesis pathway and glycogenolysis pathway. Rate limiting enzymes for the gluconeogenesis are pyruvate carboxylase, phosphoenolpyruvate creatine kinase, and the fructose 1,6 biphosphatase, depending upon various complex situations occurring in the body. Substrates of gluconeogenesis are the pyruvate, lactate, glycerol, propionic acid, any tricarboxylic acid cycle intermediate, and the amino acids. Now, in the amino acid, remember the glucogenic category 1 and the both glucogenic ketogenic category amino acids are basically the substrates of the gluconeogenesis pathway. Purely ketogenic amino acids are the leucine and the lysine. The lysine is the controversial one. Both glucogenic and the ketogenic amino acids are tyrosine, tryptophan, THREONINE, ISOLEUCINE, and the PHENYLALANINE. And all the remaining amino acid comes in the category of the glucogenic amino acids. That means alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, methionine, proline, serine, valine, all this comes in the glucogenic amino acid category. So total ketogenic amino acid are 7. 2 are purely ketogenic, that means the leucine and the lysine, and 5 are in the both categories. Total glucogenic amino acids are 18. 13 are purely glucogenic and 5 are in both the categories. So only 2 amino acids can never form glucose, rest 18 can form the glucose. Mostly 3 carbon containing compounds are a good substrate for the gluconeogenesis, for example pyruvate, lactate, alanine. Link reaction in which there is conversion of the pyruvic acid into acetyl-CoA, which actually uses the pyruvate dehydrogenase complex, is actually inhibited during the fasting state. So excess of the pyruvate which is not entering into the link reaction is used up for the gluconeogenesis. If fructose 2,6 biphosphatase is active, glycolysis decreases. Fructose 2,6 biphosphatase activity is present in the TIGAR, that means tumor protein 53 induced glycolysis and apoptosis regulator. 2-pyruvate 2-glucose formation by gluconeogenesis uses 6 ATPs, or we can say 4 ATPs and 2 GTPs. 2-lactate 2-glucose formation by gluconeogenesis uses 6 ATPs, or we can say 4 ATPs and 2 GTPs. 2-alanine 2-glucose formation by the gluconeogenesis uses again 6 ATPs, that means or we can say 4 ATPs and 2 GTPs. In gluconeogenesis, ATPs are used by pyruvate carboxylase and phosphoglycerol steps. Okay, and the GTP is used by the PEPCK, or we can say phosphoenolpyruvate kinase enzyme. So today up till here, we'll discuss about the further important notes in our next video. Thank you so much.